But thank you for talking with us today. Would you please introduce yourself?
Sure. My name is Michael Charness, and I am enjoying the fact that this is the 40th year that I've been coming to RSA. I went to my first RSA meeting in 1984 in Santa Fe.
How did you move along your path in alcohol research, and were there influences, even going back to childhood, adolescence, that led you to where you are? Were there particular people that had a special impact on the path?
So I think, like a lot of people who are at this meeting, I was curious about how things worked when I was a kid, and I loved my science classes when I was in high school. I went to college without much sense of where I was heading. I actually had to choose a major my second year of college, and I was a math major because I did well in math, and I thought, so you do the things you do well in. But after a year of that, I thought, well, so I like math, but I'm not sure I want to be a mathematician. So I kind of looked around for what else there was, and actually my older brother, who is a psychologist and was in the psychology program at McGill where I went to school, was doing that. And I thought, well, maybe I'll try some psychology. So I changed my major to psychology at that point. And there was a program at McGill that allowed you to do something more at a graduate level, that you essentially went into a lab for a couple of years and you took seminars instead of large classes. And so I ended up in that program, and I ended up doing a couple of years of research with Zalman Amit, who was looking at an animal model of alcohol self-administration, where he was trying to induce self-administration by stimulating the lateral hypothalamus. It had to do with the TIQ hypothesis of alcohol, and that something was happening with acetaldehyde and alcohol that was creating opioid-like substances, and that maybe was behind alcohol self-administration in animals. So I did that work for a couple of years. I thought I was going to go to graduate school. My last year of college, I ended up working in a crisis intervention center over the summer and decided maybe I wanted to take care of patients too. So I ended up applying to MD Ph.D. programs, and they were not in Canada. So I came to the States, and I ended up going to Johns Hopkins for medical school and started in the graduate program there as part of the MD Ph.D. program, but knew that I had 5 years of residency training and didn't want to fall 5 years behind. And so I decided I'd graduate with my class and I would do my research training after residency, which is what I did. So I spent a year in Saul Snyder's lab at Hopkins during that time. It was a wonderful experience, and it was formative in the approaches that I would take in research going forward, at least getting started.
In these interviews, I'm hearing a lot of seemingly chance encounters.
Yes.
Very impactful.
Yes.
What challenges did you face along that?
So I would say the first challenge is that after completing my residency in medicine and then in neurology, I was pretty rusty in science and really didn't have much of an idea of what I was going to do, and wasn't very good at it. It was very slow. I spent, I would say, the first 6 months trying to get an assay to work, and it worked okay. And then trying to ask a question. And you spoke about serendipity. There was this serendipitous moment, which is I was studying opioid receptor regulation in neural cells. And at that time, as I had started in the lab, Ernest Gallo gave money to UCSF, which is where I was, and Ivan Diamond, who was my mentor in the lab, was the recipient, and so we had to do something with alcohol. So we literally just, without any thought or hypothesis, I dripped alcohol on my cells and saw what happened to the only thing I knew how to measure, which was opioid receptors, and learned that it inhibited opioid receptor binding. I was looking at delta opioid receptors at the time. And that was sort of interesting, but not that interesting. But there had been work done with a similar cell line, demonstrating that if you expose that cell line to opioids for a period of time, that opioid receptor regulation changed, that there was an adaptation. And so I was curious whether anything like that would happen. Would a neural cell, in a dish without all of the influences of nutrition, behavior, social interventions, and all of the rest, would it show some capacity to adapt, maybe as a model for addiction or for alcohol addiction at the time? And what we found was that if alcohol inhibited opioid receptor binding, if you incubated the cells for a couple of days with alcohol, they started to overproduce opioid receptors. And so it looked like they were somehow sensing the reduction in opioid receptors, and something was leading to an adaptive change that happened in a cell in a dish. And that, after 9 months of puttering in the lab with nothing happened, we eventually wrote that up and it was published in Science. And I thought, well, this is pretty easy. That's how you do it. And of course, I went back into the trenches and took a long time for my next paper and so on. So I would say it started slowly. I had some early luck. And maybe the luckiest move was that after not really planning to continue in alcohol research, this serendipitous event, which is the Gallo money coming to UCSF, sort of steered me back to alcohol research. And so I went on to do that kind of work for a few more years.
How did you end up in Boston?
So that's a good question. I was introduced to my wife as a blind date through Ivan Diamond's wife, Sarah. And we actually, I had to fly to New York where she was to meet her, except I couldn't get to New York. I could get to Washington, where there was a neurology meeting, and take the train. And I met her and we got engaged that same day, and she came out to San Francisco. We got married and we had a couple of kids in San Francisco. And at a point she said, I want to go back to Boston, which is where she was from. So when I got the first 5-year renewal of an R01, I put my hat in the ring and found a job in Boston, where I was splitting my time between Brigham and Women's Hospital and VA in Boston, and started to do things on my own there.
What's that story? You talked about serendipitous finding. Have you had the eureka moments?
So eureka moments are all relative, and I haven't had very many, but the one that changed my career direction was that I was plowing away, still studying opioid receptor regulation. They finally cloned a gene. I worked with the person who did that to show that alcohol was regulating opioid receptor gene expression. And that work was starting to peter out, to be honest. I was less excited about it. I think the study section was less excited about it. And I had another serendipitous moment, which is that somebody down the hall in the building where I worked was studying bone morphogenetic proteins, which are proteins that help bone grow. But the bone morphogenetic protein, or BMP, that we were going to study had been cloned from the hippocampal library. So it was in the brain, but no one knew what bone morphogenetic proteins were doing in the brain. So we did this really thoughtful experiment, which is the same as the one I did in the Gallo lab, which is we dripped it onto my cells and stood back, watched, and nothing happened. But the next day, and especially the day after that, the cells looked very different. Instead of growing, they would divide and then separate again. They would start to grow in sheets. They were sticking to each other. And so I had no idea why that might happen, but talked to some people and started to look to see if any sticky molecules were being made. And it turned out that BMPs were inducing the genes for a couple of sticky molecules. One is called NCAM, and the other is L1. And they're both genes that are very important in the development of the nervous system and also for learning and memory. So I thought, okay, I have one more thoughtful experiment to do, which is I know that alcohol impairs cell migration. And I know sticky molecules play a role in cell migration. Now we're talking about development, about which I knew nothing. So I'm going to do this experiment, see if it alters the change in how these cells are sticking together, as they're growing in culture. And so maybe my only eureka moment was that when we incubated the cells with alcohol over a couple of days, in the presence of these BMPs, they didn't stick to each other anymore. They went back to the way they looked. Their morphology was as though there were no BMPs. So that was kind of interesting. The eureka moment was that we were testing 100 millimolar alcohol, which is a very high concentration. And we wanted to establish, a dose response curve to see what it looked like. And we went higher. And it didn't do anything more at a higher concentration, which worried us. So we decided thoughtfully to go lower. And it turned out that we were seeing effects at 1 or 2 millimolar alcohol, and the half maximal concentration was 5 millimolar. And for people who aren't familiar with this, 1 or 2 millimolar alcohol is like a half a drink. And so that was interesting because it was one of the effects of alcohol, maybe most, one of the most sensitive effects of alcohol, I guess, that a lot of the work that was being done in animals and especially in cells use these high concentrations. And here was an effect where a biological event was sensitive to half a drink, and maybe one that's important in development. So we really changed the whole direction of the lab, and we began to ask, so what's alcohol doing to change stickiness? Is it changing expression of this gene? Is it blocking its transcription? And so on. We did all those experiments. We didn't find anything. That it was the same amount of the sticky molecule. It looked like it was in the same place and everything else. So we said, well, maybe it's changing how it sticks to each other. So this experiment really just, again, was very simple-minded. You take cells and you stir them around and you see how many of them are sticking to each other. And if you did that in the presence of alcohol, it inhibited the stickiness. So was it NCAM, which was one of the molecules, or was it L1? And it turned out to be L1. And we started reading about L1, and it turns out that there is a genetic syndrome in humans where mutations in the gene for L1 caused brain abnormalities that had a lot of similarities to fetal alcohol syndrome. It eliminates or thins out the corpus callosum. It affects the structure of the cerebellum. It causes hydrocephalus and so on. And we thought, well, so maybe alcohol is a phenocopy of this disorder. And maybe one of the ways alcohol causes developmental abnormalities is by blocking cell adhesion. So we actually worked on that for a couple of decades, and we learned that alcohol was binding to a site on the molecule that affected its stickiness. And so if there's a eureka moment, there's an anti-eureka moment in science, which is that you have this great idea, you seem to have uncovered something that maybe is important in how alcohol affects the development of the brain. And then you get a call from somebody who said, you know, we saw your paper in the Journal of Cell Biology. It's really exciting. We can't get it to work. That's the other kind of moment. And so that was interesting. And we actually were finding the same thing, that there were some cells that we had cultured, clonal cell lines, that were always sensitive and others that were not sensitive. And we said, well, either this is awful, you know, we have a sort of flimsy observation, or there's something really interesting going on. And I would say for 6 months, we were really nervous because it could have been some artifact that was producing our results. But we became convinced that there were host cell differences in these cell lines that were all derived from the same parent cell line that made some of them sensitive and not others. And the fact that they literally were, you know, a few passages different from the parent meant that they were probably very similar genetically, except for something that might be different. And so we went about trying to understand what the difference was. And to make a long story short, it turns out that there are protein kinases which modify molecules that differed in their activity and their concentration in the cells that were sensitive compared to the ones that weren't. And that was true also for animal lines that were sensitive or not sensitive. And it was true also for a cohort of children with fetal alcohol spectrum disorders that we looked at from CFAS, the Collaborative Initiative on FASD. And so it looked like something was hanging together. And we eventually showed that there was an inside-out signaling event going on where if alcohol was anchored to the cytoskeleton, the sort of structural exterior of the cell, if L1 was anchored to that, it was sensitive to alcohol, but if it wasn't, then it was insensitive. So depending on where it sat in the cell, and that there were these protein kinase that determined whether or not it was anchored or not anchored to the cytoskeleton. So anyway, that was my probably one eureka moment. I had one semi-eureka moment, which was we were able to show that small molecules that blocked the effect of alcohol on L1 prevented teratogenesis in animals. So it looked like it might really be important. And at about the same time we were doing that work, Kathy Spong was showing that she could inject animals with a peptide, small peptide, at very low concentrations, 10 to the minus 15, extremely low, and prevent teratogenesis. So we said, well, does that protein change alcohol's inhibition of L1? And it did at the same concentrations. That was half a eureka. And so we did a lot of work on that peptide also and showed that it worked by changing the ability of L1 to bind to the cytoskeleton.
I really like that path, that story. Have these been explored for opportunities for potential interventions, or is it too late by the time somebody's been affected?
I get asked this question a lot, and there are two challenges. One is that the molecule appears to act at the time that alcohol is causing its effect, because alcohol is binding to this site on the L1 molecule and making it less sticky. So the question would be, if somebody came to the emergency room who happened to be pregnant, and also was under the influence of a very high concentration of alcohol, would you give them something? And of course, it's very difficult to give drugs to pregnant women because there's so much risk involved. And certainly not everybody who is exposed even to a high concentration of alcohol will have problems in their offspring. So we never did get to answer that question. Kathy showed that you could give this peptide after, not that long after, and it would still mitigate some of the teratogenic effects of alcohol.
The field of alcohol research is highly interactive and networked now and in its history.
Yeah.
What people do you consider to have had an impact on your research that was significant?
Yeah. So I'm going to go back to 1984 in my first RSA meeting. And there I was, a postdoc, standing at my poster, and Adrian Harris came over and talked to me. And I have to say, I had the experience that I hope everybody does when they come to RSA, which is, it was my first year, I was new to the field, and he was so welcoming and so cordial and seemed interested in what I was doing. I wasn't as interested as he seemed to be. When you're a postdoc, you're just not sure that what you're doing matters that much. But he was a person who was established in the field, and he actually talked to me. I thought, wow, I like this meeting. And so I mentioned him because so many of the collaborations I've had have come about from just being in a community that RSA engenders, where people are very collaborative and very cordial. And one of the things that Adrian worked on that I always followed very closely and we talked about a lot was the idea that alcohol is not disrupting membranes in a non-specific way, that even though it's a very weak drug, that there are these target proteins that it interacts with in a dose response way, and you can change an amino acid or two, as we did in ours, and as he did in so many other proteins, and demonstrate that there's a site-specific interaction. And so I always felt comfortable talking to Adrian after he came to talk to me at my poster when I was a postdoc. But I followed his work carefully because we ended up asking some of the same questions about L1. And L1 turned out to be one of those molecules that alcohol seems to interact with in a very specific way. Yeah, it's a great question. So although there have been mentions of adverse effects of prenatal alcohol exposure going back to biblical times, the recognition that alcohol causes a specific syndrome after prenatal exposure is actually pretty recent. The first mention probably was 1968 in a French language publication that no one noticed, at least not the people who don't speak French. And it was only in 1973 that Ken Jones and colleagues really described fetal alcohol syndrome in the English language literature in a couple of papers in Lancet. And so our field really begins, awareness in 1973. Now, there was knowledge of alcohol and some effects and all the rest, but to have a distinct syndrome, as was described, both by Ken Jones and David Smith, and then by Lemoyne, who is the French investigator, really set people thinking about, so this has been around for a long time, people have been drinking for a long time, why haven't we noticed this? And what do we do about it? And so I think the first historical moment was actually describing it. And that is during my lifetime. And I would say the next question that people all dwelled upon was, is it alcohol per se, or is it associated malnutrition or some other factor? And I think Kathy Sulik wrote the definitive paper on that in Science in, I think, 1981. And in that paper, she showed, in well-nourished mice with a discrete exposure at the time of gastrulation, which is about day 17 of pregnancy, that alcohol exposure reproduces the facial anomalies that help to define fetal alcohol syndrome and cause other changes in the brain as well. And she also showed that oxidative stress probably plays a role. And I would say that was one of the seminal moments in the field, and changed the way people thought about it. So, the impetus to begin to message to the public that drinking during pregnancy is not a good idea was slowed by the uncertainty about whether it was alcohol per se. But this was a moment where everybody could say, wow, alcohol really truly is a teratogen. It causes dysmorphology, it's a behavioral teratogen. And everybody went about trying to understand what really were the spectrum of effects that alcohol could cause on pregnancy. So there was a lot of work done looking at brains of animals and animal models, looking at imaging in humans, and studying both the imaging abnormalities of the brain as well as the associated cognitive, behavioral, social, adaptive changes that resulted. And that took a lot of time, but it began to produce a very clear picture that you could mimic the effects of alcohol that were observed in humans who grow up under very complex conditions in a very controlled environment using various animal models. And it led ultimately to the Surgeon General's warning that people who are pregnant or trying to conceive should not drink alcohol at all. And that was really a sea change from how the medical community viewed alcohol and pregnancy. In fact, alcohol used to be administered IV to pregnant women who were in premature labor to slow down their labor. So it changed practice. It changed the whole public health messaging. And I think it raised awareness that alcohol is a teratogen and it should be avoided during pregnancy.
Wow. I'm thinking now about sitcoms from the 1960s, classic ones, where a pregnant woman may have a glass.
Yeah.
We don't see that now.
Less. Yeah.
You've answered my next question partly already. How do you see the concepts of prevention advancing over the years? Where do you see that we still need to go?
Well, I think after the Surgeon General's warning, there were other efforts, including warning labels on bottles. It's not clear how well they work, but that's something that's done. I think one of the challenges in messaging, which exists today, is to educate women without blaming women, and to, and this is true of so much of alcohol research, to try to find a way to have this discussion without stigmatizing either mothers or children with fetal alcohol spectrum disorders. And I would say that we've made a lot more progress in understanding the pathogenesis, the mechanisms underlying alcohol's effects on the developing brain than we have in being able to convey to the public in a non-patronizing, non-stigmatizing way why prenatal exposure to alcohol is something to avoid. And I think what makes it particularly tricky, so I mentioned, day 17 pregnancy, it's a time when most women don't know they're pregnant. And there is a relatively high rate of drinking in women who are in their reproductive years. There's actually a pretty high rate of drinking even in women who know they are pregnant. So I would say that we have a lot of work to do in screening for alcohol use during pregnancy, for having effective interventions during pregnancy. CFAS, which I mentioned, has as one of its projects a prenatal intervention to try to reduce drinking. The people who drink the most are the ones who are least likely to stop drinking because they are dependent on alcohol. How do you reach that group? How do you change their behavior or give them alternatives to drinking? So there's an awful lot of work to be done. But I think the whole field of alcohol research is still challenged by how to address stigma. Somehow mental health disorders have gotten past that further than we have. There are organizations that advocate for people with mental health disorders. We have some, but they're fewer in number and their voices aren't as loud. And as a consequence, public policy hasn't really caught up with all of the risks associated with alcohol.
That's a direction where we need more work.
Yes.
How have you seen treatment developments and trials for FASD evolve over the years?
So there have been interventions that have been designed. I think one of the challenges in the field, probably somewhere between 1% and 5% of first graders have an FASD, which is a huge number. The FASD is in this country typically diagnosed in a multi-specialty group with a lot of experienced people. There aren't that many of those groups. There aren't actually that many pediatricians who are comfortable diagnosing this. And so the capacity for making the diagnosis and providing interventions is outmatched by the demand, the number of people who are affected. So that means that we can't go about diagnosis and treatment in the way that we're used to, because we might be able to diagnose well and treat well with what available treatments there are, a very small population. But that raises all sorts of healthcare equity questions, which is who gets treated. And it means that people who have less access to care will not get treated. And so one of the major goals that CFAS has is to, in a way, democratize the diagnosis and treatment by making it more available. And so CFAS is using 3D imaging, 2D imaging, machine learning, ultimately something that we hope will allow somebody to upload an image to the cloud and have come back a risk score that says, what we see suggest that there's a high risk that, you know, uh, the facial anomalies that are observed are a consequence of prenatal alcohol exposure. Um, that's only part of it. CFAS is also working. Sarah Matson is doing this. She has developed something called Brain Online, which allows people to go to a website and do a series of um, neuropsychological tests online. Again, not having to sit in a room with a psychologist, but do it themselves. Hopefully, we're going to be able to combine what's learned through that kind of information with the 3D facial imaging. It all can be hopefully integrated into a single risk score, and a person might be able to take to their pediatrician the information that they've done this thing online. They were told that they should discuss with their pediatrician the possibility that they have FASD, for example. So that would allow many, many more people to be diagnosed. But then what about treatment? And Christy Petrenko is working on some online apps, some mobile apps that allow using a program developed by Heather Carmichael Olson called Families Moving Forward, a way to mitigate some of the effects of prenatal alcohol exposure using an app-based treatment approach. So I think that's where the field needs to go, because globally, the number of people who are affected vastly exceeds the capacity of global health systems to be able to respond in kind. And there's going to have to be some way to amplify the diagnostic and treatment capacity to be able to meet the need.
What interactions do you see between research on FASD and other areas of alcohol research? Can you identify any interactions that have been key for the development of new concepts?
So it's a great question. And ultimately, we're all studying the pathophysiology of alcohol, how alcohol affects cells. The people who study FASD are interested in the developing nervous system. The people who are studying addiction are looking at the adolescent and mature nervous system. But alcohol has fundamental actions that are common to both the developing and the mature nervous system. And we've learned a lot about alcohol effects on neuroinflammation, the immune system, oxidative events, oxidative injury. That information is moving back and forth among investigators who are studying these different systems, but of course the information flows freely. And at a meeting like RSA, where it's not just a group of people studying development or a group of people studying addiction, there are people who are really looking at basic mechanisms that are going to be applicable to the developing nervous system as much as the mature nervous system.
For researchers at the start of their careers, do you have any advice to offer? There are basic scientists, clinicians, physician scientists, biochemists, people focused on psychosocial aspects.
So my advice is, we really need you. This is an enormous problem to study. And it really will take, not a village, a metropolis of investigators who are able to look at every aspect of the problem that you mentioned, from very basic biological understanding of mechanisms to the people who are involved on the treatment side. We all have to talk to each other, but we need to repopulate and expand the field so that there are more and more investigators, which means we need you. We need you to come to the meetings, to be inspired, to work with the populations, and to help us provide better treatments, have a better understanding of what we should be targeting to treat. Because the problem isn't going away. People are drinking at higher rates ever since the pandemic. People are dying at higher rates than ever as a consequence of alcohol's effects on so many organ systems, including the brain. And we just need people to come into the field and to stay with it. You know, you have to be passionate about research to do this. We know that a lot of people are taking their research training to industry, but industry ultimately depends on the findings of the people who we see at this meeting to ultimately have impact and change the effects of alcohol on people, whole people, organ systems, molecules, cells. So stick with it, stay with us. We really need you.
For researchers in general, do you have anything that you would like to ask from all of us?
Just what I said, you know, it takes more than a village to study something as complex as alcohol and its protein effects on the human body. And keep coming to the meeting, keep sharing your research, and don't just stop at the posters in your own field, because the one that's going to change your future as a researcher is around the corner in an area you haven't thought of.
For members of the public who may watch this video, is there anything that you would like to communicate directly to them?
I would say be patient. We have treatments for alcohol. You should be asking for them if you have a problem with alcohol use disorder. It's a small percentage of people who have an alcohol use disorder who have access to some of these treatments. Part of that is, I think, an unwillingness of some clinicians to prescribe them. There's, I think, an ethos in the field that goes back to the earlier days when the idea of using a drug to replace a drug was not popular. I think that philosophy still is strong in some camps. I think I mentioned health equity, healthcare equity. We should be thoughtful in a country where we have such poor health care equity, how we can make healthcare available to people who don't have very good access because they can't afford it, or they are not treated with the same respect for their needs as other people. And I think everybody has a role to play in that. That's really for the public and for the policymakers to be able to change. If we had the perfect treatment, we wouldn't necessarily be able to solve the problem, because people need access to care and to treatments. So I think that's everyone's responsibility.